Now,
in molecular biology and genetics, translation is the process in which cellular ribosomes create proteins. It is part of the process of gene expression. In translation, messenger RNA mRNA produced by transcription is decoded by a ribosome complex to produce a specific amino acid chain or polypeptide. The latter will fold into active protein. In bacteria, translation occurs in the cell cytoplasm, where the large and small subunits of the ribosome are located and bind to the mRNA. In eukaryotes, translation occurs across the membrane of the endoplasmic reticulum in a process called vectorial synthesis.
I think we're going to have to re-record.
No, you.
Ribosome facilitates decoding by inducing the bindings of tRNAs with complementary anticodon sequences to that of the mRNA. The tRNAs carry specific amino acids that are chained together into a polypeptide, which, as the mRNA passes through and is read by the ribosome in a fashion reminiscent of that to a stock ticker or a ticker tape. In many instances, the entire ribosome mRNA complex binds to the outer membrane of the rough endoplasmic reticulum and releases the nascent protein polypeptide inside for later vesicle transport and secretion outside of the cell. Many types of transcribed RNA, such as transfer RNA, ribosomal RNA, and small nuclear RNA, do not undergo translation into proteins.
Translation proceeds in 4 phases: initiation, elongation, translocation, and termination, all describing the growth of the amino acid chain or polypeptide that is the product of translation. Amino acids are brought to ribosomes and assembled into proteins.
The following factors must be present for DNA replication and transcription: gene to act as a template, supply of free RNA nucleotide enzymes, ATP. The base sequence in DNA molecule represented by the letters A, T, C, G make up the genetic code. The bases hydrogen bond together in a complementary manner between strands. A will always go with T, U in RNA, and G will always go with C.
These are the key points. Number 1. DNA directs the production of proteins, but it does not directly produce them. For protein synthesis to occur, 2 things must happen: transcription and translation.
RNA is chemically very similar to DNA, except for the sugar component of RNA is ribose instead of deoxyribose, and contains the base uracil in place of thymine. The RNA is transcribed from DNA for the purpose of protein synthesis is called messenger RNA or mRNA.
Number 3. When proteins are formed from mRNA information, it is called translation. Translation occurs in the cytoplasm of prokaryotes at a very specific protein RNA complex called a ribosome.
Number 4. The process of transcription and translation occurs in all organisms. While the overall process is similar, the lack of a nucleus makes this process somewhat different in prokaryotes and eukaryotes.
Number 5. In eukaryotes, transcription occurs in the nucleus and the translation in the cytoplasm. The mRNA produced from transcription is called pre-mRNA or primary transcription. The pre-mRNA is altered slightly before it leaves the nucleus for translation.
And finally, the process of DNA, RNA, proteins is commonly referred to as the central dogma, a phrase introduced by Francis Crick in 1956.